two roles of water within the plant aside from participating in chemical breakdown reactions. Transpiration, transpiration cooling, the movement of mineral ions, those are all acceptable responses. The mass flow through of sucrose through phloem tissue. Excuse me. The mass flow. Those are both acceptable responses. And that's how Unit 4 can be tested in a Unit 1 question. Unit 1 can be tested in a Unit 4 question. And Unit 1 can be tested in a Unit 3 question or a Unit 2 question. They often mix and match the units. Enzymes are globular proteins. What is meant by the term globular? And that is worth 2 points. You must describe the term globular. You must define globular. So let's do that now. We're doing that now, and that is here. Nope, that is here. There are 2 types of proteins. The 2 types of proteins, for your purposes, are fibrous and globular proteins. Fibrous proteins are fibers, like silk, wool, and skin. They are generally insoluble in water, in acid, in base. They have strong intermolecular forces that hold them together. And their structure is typically sheets, threads, or helices. They are threads. They are what bind us, like the force. So they have very strong forces. Globular proteins are things like albumin, which is found in egg. They are found in food. They are proteins like hemoglobin, myoglobin, insulin hormones, peptide hormones, and the like. Things that are soluble in water, in acid, and in base. If they are soluble, they are much more likely to be globular. They often will have weak intermolecular forces that hold them together. What is the difference here? Why is fibrous strong and globular weak? What does that mean in terms of reactions? It comes down to denaturation. Globular proteins are easily denatured by raising temperature, by raising salinity, which is salt content. They are easily denatured by raising acid or lowering acid, raising base. Changing the environment of a globular protein will change the 3D conformational structure of a globular protein. Whereas in a fibrous protein, they are meant to be tough, to take a licking and keep on ticking. They do not break under stress. However, burn them, and it will burn. Hair can burn. Hair can definitely burn. Wool can definitely burn. Skin can definitely burn. You can create incredibly difficult, dangerous environmental conditions that destroy fibrous proteins. Strong acids, strong bases, strong heat, strong cold that crack and break these intermolecular forces. You must have stronger forces to break fibrous proteins than you have globular proteins. That is why this is strong and weak. That is why it matters. Strong intermolecular forces means environmental changes mean a lot less to a fibrous protein. A wool sweater will not shatter unless you dip it in liquid nitrogen first. But a globular protein might under the right cold condition. Like lower the temperature enough and then hit it with enough concussive force, it might shatter. But wool, no, it ain't gonna happen. So that's why strong and weak intermolecular forces are in play here. That's why these shapes matter. Form equals function. So the structure of a globular protein is folded, ball-like, three-dimensional. In a fibrous protein, it is a thread, a strand, or a sheet. Flat, 2D or 1D. But not really. It's really still 3D, but you know, for our purposes, it's close enough. So that is globular. Globular, like a globe, round. And it results from weak intermolecular forces and solubility, mostly due to hydrophilic and hydrophobic interactions that's not covered in this slide. This is hemoglobin, our model globular protein, a quaternary structure made up of multiple tertiary structures. It happens to have 4. It could have had 3, but it doesn't, so, you know, it's still a quaternary structure. This is what it looks like on a bad hair day. Globular proteins like hemoglobin will typically have a job to do. Hemoglobin's job is to carry oxygen in the blood, as we've discussed in the past, as we will discuss again in the future. Carbaminohemoglobin is a compound of hemoglobin and carbon dioxide. It is attached to the terminal amino ends of hemoglobin. I've corrected that in the past. 10% of carbon dioxide can be carried in the blood in this way. Carboxyhemoglobin is carbon monoxide and hemoglobin. That is attached directly to the iron-containing heme subunit, the prosthetic group. Prosthetic groups, which I haven't gone over, by the way. Prosthetic groups, as a quick aside, are groups that are not protein, that are attached to a protein. So a carbohydrate that is attached to a protein is a prosthetic group, an organic prosthetic group, because it's made of carbon. A lipid attached to a protein is a lipoprotein, and that is an organic prosthetic group because it has carbon. An inorganic prosthetic group is something like heme, that is primarily, the active part of it is not carbon. Iron is the important bit here. That is a non-organic, an inorganic prosthetic group. So that is the important part here. So it's thanks to the iron. So, oh, I could have waited a slide. Forgot about that. So organic prosthetic groups could be vitamins, sugars, or lipids, and inorganic could be metals or ions, or metal ions and such. But it's not amino acid. Protein attaching to protein is nothing new. That's just a tertiary structure. That's just a quaternary structure. Protein attaching to protein via peptide bonds is just primary structure. With hydrogen bonds, it's secondary. With hydrogen and other bonds, it's tertiary. So when you have tightly bound, it might just be bonded by linking it by hooks or whatever, or it could be actually covalently bonded to a protein. Doesn't matter. It doesn't matter. It's still a prosthetic group. It's still not a protein attached to a protein. And coenzymes we'll discuss another day, or probably never. We may never discuss coenzymes. It may never become important. Let's talk about fibrous proteins. Collagen. Collagen is important for the Cambridge exam. And this is the only time we'll talk about it, because hemoglobin we'll talk about again. So collagen you gotta learn now. Collagen is a tripeptide. It is a quaternary structure composed of 3 polypeptide chains. That is the individual polypeptide unit. It is a group of, the collagens are a group of naturally occurring fibrous proteins most abundant in mammals. It acts as the glue that holds the body together. It shows very little in the way of tertiary structure. It has very little 3D conformation because it's just a strand. It's simply a straight strand. And because it's a straight strand, it's not really 3-dimensional. But it's still a quaternary structure because it has more than one chain. So the collagen tripeptide is the individual polypeptide itself. It's the tripeptide is the quaternary structure. When you combine multiple of them together, you get a collagen microfibril. You must learn these words, and you must learn the order that they work in, because they can get you points on Cambridge. A collagen microfibril is multiple collagen polypeptides together. A collagen fibril is multiple microfibrils together. Multiple fibrils together forms a collagen fiber. Microfibril, fibril, fiber. The same idea as cellulose. Microfibril, fibril, and fiber. It's the same organizational structure. It's the same. Smallest individual strand to group of individual strands, to group of individual strands in a group, groups of groups, to groups of groups of groups. I am group. So collagen fibers, these are thick cords. They are hard to break. They are strong. You make violin strings out of this stuff. You make bow strings out of this stuff. The chordae tendineae in the heart is made out of this stuff. Your Achilles tendon is made out of this stuff. And when you have weakness of this stuff, you get diseases like Ehlers-Danlos, where you have poor collagen and it allows the stretch without that tension, without that, it allows it to snap back, but it doesn't allow it to stay, stiff and stable. Collagen makes it stiff. It makes it not stretchy. It makes it hard to bend and stretch. Scleroderma is scar tissue of this collagen, which does not stretch and bend, and it is tight and taut. So, elastin is another type of fibrous protein. Elastin is another type of fibrous protein that you will learn more about, and have already learned more about, that allows that recoil from stretch back to original shape. It is another fibrous protein that allows the recoil. And if elastin is broken or doesn't work, then you have no recoil, and whatever stretch happens stays floppy. So Ehlers-Danlos is where elastin is working. Collagen is not. You have the recoil, but you can stretch. There is no rigidity. There is all stretch and recoil. So those collagen's job is to be connective tissue, to be supporting tissue. That's homework. stuff will go. Nucleic acids. Now we're on to nucleic acids. The basic structure of a nucleic acid. Is a phosphate group, a sugar, a pentose sugar, to be precise, 5-carbon sugar. It can be ribose or deoxyribose. And a nitrogenous base. A carbon with there's some carbon in here, but there's primarily nitrogen involved. There's a lot of nitrogen going on in here. But look, carbon, 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 carbon. There's carbon wherever there isn't nitrogen in all this ring structure. So this is a nitrogenous base. It still contains carbon, but there's a lot of nitrogen up in here. You need a lot of nitrate to make this, or ammonia. So this is the basic structure of a nucleic acid. This happens to be adenine. So your DNA has cytosine, guanine, adenine, and thymine. Your RNA has cytosine, guanine, adenine, and uracil. Thymine belongs to DNA, uracil belongs to RNA. They will never, ever, 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 ever have thymine on RNA. You will never have uracil on DNA. That is how you tell it apart on the Cambridge exam. If you are asked a question like, why is this structure RNA, not DNA? And you have to look at it, and one of them is labeled uracil, because the structure contains uracil. Uracil is only found in RNA, and this contains it. DNA does not contain uracil, and this structure has it. DNA only contains thymine, not uracil. If you see uracil, you have to mention that you see uracil, and uracil is only found in RNA. You will not get a point for stating that only thymine exists in DNA, and that's why. You don't see thymine in a picture that only contains uracil. You have to comment on what you see. If you mention thymine, you have to also mention that you see uracil here. And in DNA, uracil is, thymine is there instead of uracil. You have to mention what you see in the image on paper too. If you don't go based on what you see in the picture, you cannot get full credit. Sometimes you can't get any credit at all, especially if they say something like, using the figure whatever, using figure 1.1 or 2.3, or in reference to figure blah, blah, blah. Explain why. If you do not reference the figure, you cannot get maximum points. Do you understand this?